It's an enormous pleasure and an honor for the Institute of Advanced Studies of the University of São Paulo to have Professor Luisi with us again. Professor Luisi is probably the scientist in the world who is best known in the field of origin of life for a long time. He has made extremely important contributions to understand what he will explain. I will not try to do that. And he has published a lot, and many people have studied and enjoyed his company. But I want to mention two other things. Professor Luisi worked for a long time in Switzerland, and now he's in Italy. But he's not only a scientist in the very 21st century sense of the word. He is a man from the Renaissance. He's a humanist, and he's responsible for one of the most important moves in the world to unite scientific thought with humanistic and spiritualistic thought. And he has been responsible for that for many, many years. It is an honor to have him here again. I think we will have him here next year again, and etc., for the next 100 years. Today, he will speak about emergence, but not only of life, but of life and the meaning of that emergence to the emergence of consciousness. And it's a pleasure to listen to him, and he's a good friend, and we hope to have him with us anytime, at any place he decides. He makes his own choice when he's coming here. Thank you very much.
Thank you. The pleasure and the honor is mine, and I'm very happy to be again here in this place, in this country, where I count now many friends. And just because we are among friends, I decided to give you here a kind of very broad spectrum, showing to you how research on origin of life is linked to the more general field of biology, synthetic biology, and then from there a little step ahead towards cognitive science, and we even talk at the very end a little bit about consciousness. So that's why this kind of ambitious title, from the emergence of life to the emergence of consciousness. But indeed I believe that this arc, it's possible in a scientific way, so without new age easiness and facilities, but still keeping to the language of science. And I hope you will follow that with me. So let's start where science starts, when science goes at the very beginning. And science at the very beginning likes to put, of course, the Big Bang. This is the origin of everything we have in our universe, including all laws of nature, including time, energy, space, all comes for our modern science from this little spot of supercondensed matter. You know how the story goes. Our universe is expanding and expanding, so if we go back in time, then we find the contraction and we find the point in which everything originates. And according to the incredible narrative of the Big Bang, which is really a kind of a narrative which is similar to, in a way, to the narrative of the old mythological stories about creation. The thing exploded 13.7 billion years ago. There was not, however, talking about explosion and about something exploding in space is already wrong, because there was no space to explode into. You know, space, time were created at this very point. And already after 100 of seconds, our science is pretty precise on that. This little thing which was rapidly expanding now had the temperature of 100 million degrees and the density which was 4 billion times larger than water. You know, they're really numbers which are beyond our comprehension. Important for our talks here is the formation when the temperature was, a little cooler, a little lower. The formation of these two elements, hydrogen and helium, from which all what we have now in the universe originated. In a proportion at the time of the Big Bang of 3/4 to 3 to 1, 3/4 to 1/4, which is still the proportion of today. Very little is changed in this proportion. Important is the beginning of the gigantic cold clouds, from which, when the expansion proceeded and the temperature cooled down, we had the formation of the first galaxies, where millions and billions of stars condensed together in what today we have as very beautiful things to observe from our telescope. Incredible aggregation of stars. This is called by the astronomer the fingers of God. They have all this kind of and here I mention here the very famous question of Heidegger, voiced before him by Leibniz and Spinoza, and one question to which we scientists are mute. We cannot give any answer. And the question is, why all that, instead of nothing? Which is a question which reverberates through the centuries and also from the antiquity, and which brings us to the point where science stops and where perhaps philosophy and spirituality begins. This very. But more important for the talk today is that after all this time, about 5 billion years ago, our mother cloud originated, from which the solar system came out. And here we have the formation of the first molecules, and from these particles then the planets and everything we have now originates. And we assist then to the, if these things, let's see, works, to the formation of the Earth, which at that time, about 4.5 billion years ago, was a kind of fireball, so very inhospitable for all kind of life we are thinking of today. Kind of place for volcano eruption and all kind of geological accidents. But soon enough, you see the change in color. The Earth began to cool down, and then the first endogenous reactions took place on the crust, and also from the space there was a lot of bombardment and a lot actually of carbonaceous material began to arrive on our surface. So that after this time, so we are about 4 billion years ago, our crust was full of carbonaceous material, and there was enough then material to start the soft organic reactions which then were supposed to bring our life. And after that, of course, indeed, life. Let's see what happens here. Life began to form first in the sea, and later on, of course, also on land, up to the formation of mammals, life as we know now. So the question for science is very simple. The Earth life was not there. There was a fireball. After a few 100 million years, the Earth was full of life. How this transformation came about? In terms of geological time, we have the formation of the first cold rocks about 3.9 billion years ago, and we have another important date, the age of the first cells, and we all agree that about at this time, 3.5 billion years ago, our Earth, our planet was full or had a lot of microbes, so very simple unicellular organisms, so that we have these windows to go from non-life to life about 400 million years, but most of us believe that since here we had already full-fledged cells with the old genomes, the origin of life was before, and so that the windows of origin of life is some few million years in between, perhaps 3.7 billion years ago. And how this transformation took place? Well, according to modern science, life on Earth arose from the inanimate matter, from those simple molecules that form the crust of the Earth, endogenous material or material coming from the space, through a series of very long series of steps of increasing molecular complexity. And the increase of molecular complexity was attended by an increase of chemical functionality, up to the point where the first structure which were capable of making copies of themselves, so the first protocells, originate life. And according to the vision we have, the prebiotic atmosphere, prebiotic means before life, contain only these four gases: water vapor, hydrogen, methane, and ammonia, which, due to the energy coming from lightning, combine together to form the first amino acids. And you can say, well, this sounds like fantasy. Well, this was indeed present in the first book on the origin of life, written by a Russian chemist, Alexander Ivanovich Oparin, wrote this book in 1924. The book was ignorant, was not known for several years in the West, until it was translated into English and fell into the hands of a very young American, brilliant scientist who was doing his doctorate in the laboratory of Professor Urey, a Nobel laureate in Chicago. And he fell in love with this idea of origin of life and asked his professor to let him work on the field. The professor said, no, of course, this is waste of time. I give you 1 week if you want to amuse yourself with this nonsense. And this week that Stanley Miller took and accepted changed really our perception of life science, as Stanley Miller put in a flask the 4 gases that, according to Oparin, were forming our prebiotic atmosphere, and put 2 electrodes with electric discharges in order to simulate the lightening of the prebiotic time. And that was it. And after a relatively short time, he saw that the walls of this flask were becoming brown. It was a kind of powder, and the analysis showed that this powder was made out of rather complex molecules, amino acid like glutamic acid, aspartic acid, alanine, glycine, and other important molecules of biological importance. So all from very simple gases, from a very simple form of energy. And this, of course, was the beginning of the experimental evidence that indeed life could originate from very simple initial molecules. Was the beginning of prebiotic chemistry, was 1953, the same year in which Watson and Crick discovered the structure of DNA, very important year for life sciences. So here the idea of that increasing complexity little by little, of course amino acid, we are still very far away from life, but the idea that there would be possible to increase molecular complexity to go up to the cell was fortified certainly by this famous experiment. This is the original flask of Stanley Miller, and this is the kind of arbitrary scale of complexity. But before proceeding, let's give a short answer to a very big question. What is that? What is that science define as life? As this, of course, it's a very hairy question, as particularly in our civilization, where the notion of life is linked to the notion of soul, and but also is linked to poetry, literature, and mountains are living, and Gaia is living, and the lake, the crystals are. There is a lot of confusion. So then, in this confusion, what is that science can say precisely about the question, what is life? There are many schools, and answer at the best is, however, to take a kind of phenomenological approach and go there to tackle this question where life has its simplest possible expression of life, in the world of unicellular microbes, those things which are only few micron. A micron is a 1,000 of a millimeter across, and so the simplest possible form of life. Let's see what life is here, and then let's escalate up. Simple things, yes, but if you look inside with the zoom of a biochemist, you are terrified by the complexity inside these little microbes. What you see here is this spaccato of the metabolic network of E. coli, which is a microbe in our intestine, and each point is a chemical compound, and each line is a chemical reaction, and each chemical reaction is catalyzed, namely made possible by a large specific protein, an enzyme, so that we have in these thousands of reactions connected the one to the other. You know, this is part of this cycle, but is also part of all the others. With the working of thousands and thousands of enzymes. So, an incredible complexity. What do we do? Well, the scientist, when has this difficult problem, has a trick. The trick is, let's make a model. A kind of cartoon which captures the main feature of the complexity. So yes, the cell is complicated, but this microbe finally is a compartment, here in cross-section, spherical compartment, defined by a membrane which separates the inside from the outside, which is semi-permeable as it lets some nutrients come in and some other stuff goes out. And inside there are all these reactions. Well, what do we learn from this cartoon? We learn a good deal about life. First of all, we notice one thing, that despite these thousands of transformations that take place in a microbe, the microbe remains always itself. A liver cell remains always a liver cell, and a yeast cell remains an yeast cell despite all these transformations. So how is this contradiction possible, that you have a lot of changes, transformation, and then you have always the same structure?
Do you want me to change the slides?
No, okay, thank you very much. In fact, that's the first discovery about life. The cell's main activity is to maintain its own identity, what we call self-maintenance, despite this enormous number of transformations. How is that possible? Is it possible because the cell remakes from within all those things which are transformed away? There is a process of regeneration from within, from the internal structure of the cell, that remakes all ATP, all glucose, or all proteins which are transformed away. And of course, we immediately think that what we say about the microbe is also true for an elephant. An elephant remains always itself, at least for the time of the steady state, for a certain time of observation, despite the fact that the elephant is the place of thousands, maybe millions of changes and chemical transformation. But all the hemoglobin of the elephant, which is destroyed catabolically, is remade from within, and the elephant, or we, lose millions of skin cells every second, but these are remade from within, and I shave every morning, but the morning afterward there is again, and my muscle decrease in size if I don't eat, but so life can be seen as a factory that remakes itself from within. That we learn from the phenomenology of the microbe. And there is a second observation which is equally important and surprisingly so. If you look at our cartoon and ask the question, yes, this is the life of a microbe, but where is life localize is maybe this reaction or this point here, or what is life? Well, we learn immediately that life is everything. Life is not localized in any spot, but life is a global property. Is a distributed quality. Is not due to a particular center, any particular compound or any particular reaction, but is a global distributed quality. This notion that you have a collective something which is self-organized and working, but has no center of localization, has no director, no cabin of command. It's very important in modern theory of complexity. If you think of our brain, where is the brain localized? Brain is everything, all brain. But also if you observe a beehive or a termite nest, where is the termite? It's of course the whole complexity. But if you look at a person, where is the Luisi localized? Is the lungs, is the brain, is the heart? Of course, the self is the emergence of many things together, is the integration of heart with lungs, with arteries, with veins, with bones. It's the congregation of all many things together without a center of localization. There is no point here that you can point as this point is life of Luisi or the elephant or any. So that this notion of that you have a distributed quality without a center of it's very important, and you can arrive at the general observation that life of a cell, but also of the elephant, is defined not so much by the local structure of the components, but by the general organization. Life must be interpreted in terms of relations among the components, lungs with heart, with kidneys, with liver, with brain, with arteries, and with memories, with feeling, all together, so that this is part of the general theory of life told by Maturana and Varela in their autopoiesis. And in fact now a couple of citations from them to reinforce this notion of life, so that life, living system have this capability of transforming inside themselves. So there is a self-organization which is capable of giving products, and this product is the organization itself. And another equivalent in a recent interview, Maturana has to say, when you regard the living system, you always find a network of processes or molecules that interact in such way as to produce the network that produce them, autopoiesis, and that determine its boundary. And all, without a center of localization, you have a network of processes that in your own life you can see as the processes that connect liver with brain, with lungs, with heart. All these processes that have as a product what? The organization of the living itself. In a cell, this is more easier to see, and each living organism has also another apparent contradiction. It's open from the thermodynamic point of view, as you have to receive energy and food. On the other hand, is, as we say, operationally closed. Operationally closed is an epistemology notion that simply means that all information to make the elephant is contained in the elephant itself. The elephant does not need any information from outside to be an elephant. It's open because it has to eat and has to excrete something, thermodynamically open and operationally in terms of information closed. So this we learn about life, and you can have this as a general definition that life is self-maintenance from within, so the maintenance of the identity due to this dynamic network of interaction, and this network is auto-poietically produced in the sense that are defined and constructed by the system itself. And if you want another definition that we have learned from the study of this macroorganism extended then to the elephant, we can say that life is a system spatially defined by a boundary made by the system itself, as our skin and the membrane of a bacteria is made by the inside, and system which is self-sustaining by regenerating the system component from the inside. And all this... Okay, so I don't know, if you have questions about this first part, we can discuss them. Otherwise, I go to a second, to another part, which is we keep short and connected with some philosophical questions, and then we'll come to some experimental research lines. So the philosophical question I want to confront you with is one which is still very much debated in modern meetings on the origin of life. And this is the question, okay, let's accept in terms of science this increase of complexity going from atoms to molecules and so on till cells. In this pathway of increasing complexity, something which is an obligatory pathway, namely that there was no other way, namely is the origin of life was an obligatory process, had to come, had to exist, or opposite view is the product of chance, this kind of origin of life. And also in a recent meeting that I have organized in San Sebastian in Spain, this question was still there, and there are people who belong to the party of determinism. Determinism means generally that you have a series of steps, and each step determines causally the next one and is determined causally by the previous one. So you have a series of events linked to each other by cause-effect. And in the case of origin of life, you see the sentence of Christian de Duve, a Nobel laureate, who says, I favor the view that life was bound to arise, had to arise, under the physical, chemical condition that surrounded its birth. And Morovitz says the same, and then there is the famous sentence by, again, Christian de Duve, who in his book writes, it is self-evident that the universe was pregnant with life and the biosphere with men. So the idea that the origin of life was an inescapable event. In fact, this view of Christian de Duve is often referred to in the literature as the gospel of inescapability. This is the deterministic view, and the opposite view, which is favored by the majority of scientists in the field, or apparently so, is the view of contingency. Contingency is the fashionable word for chance. Once upon a time we used to say chance. Contingency is more refined, and indeed it's a higher level of sophistication, as contingency has to do with the simultaneous occurrence of events or factors or parameters which are independent but occur simultaneously to determine a certain effect. For example, let's do a very simple example. I'm walking on a trottoir and a tail comes from the roof and hits me on the head. And I say, oh, what an unfortunate chance. Is an accident, is a chance. It is chance, it is an accident. However, this depends on many simultaneously occurring independent factors. Depends on the velocity by which I was walking. A little slower, a little faster, this would not have happened. If the roof would have been made new, the tail would not have fallen from the roof. If there would be a lot of wind, or if the density of the tail would have been different, if, it so happened that all these if conjure together to determine that accident. But notice that each of these parameters is per se deterministic. You know, the tail falls because the roof is old, and it's a kind of deterministic reason that something falls from something which is old. And this contingency notion is very important in the finances, and you always find this word if you look at papers on the market. And but anyway, in terms of science, life science is extremely important when you consider all the problems of evolution and origin of life. And here I give you the example of J. Stephen Gould, who was perhaps the main modern proponent for the theory of contingency, a wonderful writer, among other things. In his book, The Wonderful Life, who won a Pulitzer Prize of Literature, he considered the, let's see what I've done with my pointer, probably in my pocket. Yes, he considered the origin of life from 3.5 billion years ago to now, and he points out that over 2/3 of this life on Earth was occupied only by unicellular organisms, that only about 1 billion years ago there was the so-called Cambrian Revolution, with the invention of sex, meaning intercrossing of DNA among the various unicellular organisms, with the invention of the multicellularity, creation of the first algae and fish and mushrooms, etc., and the beginning of primates, mammals, mankind. And he says, this Cambrian revolution that shaped us is or was certainly the result of a series of contingent parameters: the temperature, the salinity, the pH, the pressure, the concentration of reagents, the location. Change only one of these parameters makes so that the temperature would have been 5 degrees higher, or the salinity 10% lower, or the pH 2 units lower. This Cambrian revolution would not have happened. He says, run the tape again, a sentence which became famous in the life science, meaning, do this experiment again. And the first step from here to here might have taken 12 billion years instead of 2. With the consequence, notice that then mankind may have not been here. And this is one of the bitter pills of contingency, because somebody say, then we are the product of chance. To say we are the product of chance is a little too extreme, but certainly if we accept this idea that all what happens in the history of evolution is due to contingent parameters, to something that happened just because the simultaneous juxtaposition of independent parameter, here temperature and pressure and salinity, then yes, then the inescapable conclusion would be that we might have not been here, which is something that many people don't like, and actually give rise to a lot of controversy between science and religion. Contingency is a very bad pill to swallow, and even if you ask scientists who believe in contingency, and you ask, then you agree that mankind might have not been here, then there is a lot of excitation to say yes. It's because emotionally it's a little difficult to accept. And in fact, Jacques Monod, another proponent of contingency, and with his famous book Chance and Necessity, a bestseller in the early 70s, writes, we would like to think ourselves necessary, inevitable, ordained for all eternity, or religions, philosophy, even part of science, testify of the unwearying, heroic effort of mankind, desperately denying its own contingency. We don't want to be the product of chance, and the literature, you can spot many ways by which there is this kind of denial of our own. Let's see some experiments connected, however, to this question. See where science and philosophy meet, and let's see whether we can do some experiments, some research project on this very point of the contact dichotomy between absolute determinism and contingency. For that, consider the proteins of our life. And proteins, you know that better than I, are chains of amino acid, usually linear sequences, where each of these balls is an amino acid, and in principle each of them can be 20 different things. So if you consider a chain long 60, then, and you ask, here are some proteins, here is an lysozyme, here is an antibody, but if you consider one long 60, and you ask your students, tell me in how many ways can a chain with 60 amino acid residues exist? Then the answer is, well, you can have 20 possibilities in the first position, 20 in the second, 20 in the third. Then the answer is 20 times 20 times 20, which is, according to some simple calculation, corresponds to the number of atoms in all universe. The proteins we have on our Earth and that form our life, our proteins, are only citation by a few thousand of billions, 10 to the 12, 10, 14. And then this ratio is something that is very much telling, because this is the ratio that exists between the whole Sahara Desert and one single grain of sand. Or if you want, is the ratio between the whole known universe and the space occupied by one hydrogen atom. So we live on one grain of sand out of the possibility, theoretically speaking, of the entire Sahara Desert, which elicit a series of interesting questions from the scientific point of view. How have these few proteins of ours been selected out? Maybe they have been selected out because they have special physical or thermodynamic properties like solubility, stability, folding. Maybe these few billions proteins we have are the only one which can be stable or soluble or folded. Well, then, if it is so, let's or the other possibility is that they are not special at all, but they have chosen out of chance. Then let's do them, synthesize in the lab proteins which are not existing in nature, that we call never-born proteins, and compare the property of these never-born proteins, which have never been exposed to evolution, with our proteins, the protein of life. This is one of the projects in my group, and how to make never-born proteins. It's conceptually very easy. You know that protein comes, according to the normal protein expression in ribosomes, from a string of DNA. So a certain string of DNA gives rise to a protein. Then you design first by computer a DNA, totally theoretically, asking your computer to mix randomly the 4 bases, adenine, thymine, guanine, cytosine. And if you do a random DNA, the probability that you hit a DNA which exists in nature is almost 0, because it's the ratio 10 to the 70 to 10 to the 12. So then you ask a company, and there are many now, to make for you a library of totally random DNA, and the company deliver you a reaction vial containing 10 to the 13 sequences of DNA which doesn't exist in nature. You put them in bacteria and they produce proteins. And so that things is conceptually simple. We use this kind of phage display. This is a phage, a virus, which contains filamentous DNA, and in here with certain procedure we insert this foreign DNA, which does not exist in nature, which produce a protein, and then this phage is injected into bacteria, which produce billions of this phage, which with their proteins. And so this protein is our never-born protein, which you can isolate because there is a tag for antibody binding, and there is a little piece for studying the folding. But the basic point is that you can, then determine the primary sequence of these proteins, and here are some of them, proteins which are created in the lab and never seen by nature before. Maybe a couple of them have been seen and then forgotten. Some of them we have purified and studied by circular dichroism and fluorescence. But the main point is that these proteins are, look like our proteins. Those which have been studied are soluble and folded, thermodynamically stable, thermoreversible, so that the conclusion, the tentative conclusion, is that since that our proteins, the protein that make our life, have nothing special from the point of view of thermodynamic stability, folding, solubility, and we may be tempted to say perhaps then the proteins of our life, since they have not been chosen because of their thermodynamic quality, then maybe are just the product of chance. You may not agree, some people do not agree, but the point here is to show that with experimental research you can touch this point of philosophy and then join experimental research with philosophical question. And another approach of also synthetic biology, which is that part of biology interest in synthesizing alternative structure or form of life, is connected with the question, it is possible to reproduce in the laboratory life, of course, restraining ourselves to cellular life. Just a couple of slides on that. Talking about cellular life, synthetically, we mean the minimal cell, of course, not a cell like Escherichia coli with over 2,000 genes and several thousand enzymes, but we mean the simplest possible living cell, namely one that contains the minimal and sufficient number of components to be defined as alive. Alive, as we have defined before, with self-maintenance, and then in addition the capability of making copies of itself, and perhaps the capability of evolving. And for that, in the research, we use this compartment vesicle made by lipids, so we call that liposomes. We contain water inside, water outside, and this is the bilayer structure that form also all our biological membranes, so the same here and here. That's why they are good models for cells. And you see here under the microscope some of these giant vesicles, giant because they're big enough to be seen by optical microscopy. You see it moving and dancing, they look like alive, but they're just water bubbles. And here again the project of in which we take this liposome and we put inside enzymes and DNA, but in the minimal possible amount, so to create a formal life which is the simplest possible. And we are together with others in this project. We started by putting inside the component for molecular biology, starting with the polymerase chain reaction. We are already in the middle 90. Then we could put inside the ribosome to make inside this compartment the first proteins. And now we and many other groups are putting inside all the ribosomal components to make proteins. And actually we work now with a kit of enzyme of only 37 enzymes. It's a particular kit developed by Ueda in Tokyo and other macromolecular components which permits the expression of proteins. And we put that inside the liposome, expressing then proteins. And this is the expression of the green fluorescent protein, which we choose because it's green and for detection reasons. And so that inside these little things we have all the function that permits the production of proteins. And now we are trying to extend this system in terms of complexity to have these little cells that are capable also of reproduction. And this is the field of synthetic life, which is very fashionable nowadays and maybe important, both for demonstrating that life is, after all, a chemical property, with due respect for all the complexity that accompanies these words, and also may be important for therapeutical reasons, which we don't go to. Now, the last part, if I still have 5-7 minutes, it's the steps, which is very important to go from this kind of biology to human level. And for that I have to add something which I've not done until now, because we have defined until now life in terms of the structure of this autopoietic machine, the cell, forgetting the interaction with the environment. And of course, we cannot talk about life if we don't take into account what is around us, because what is around us brings us food and energy. And this is so true that the Maturana and Varela, the two of the Santiago theory, Santiago because they both were from Santiago de Chile, go so far to say that we cannot talk about life if we don't talk also with interaction with the environment. Interaction of a very specific nature that they call cognition. Maybe not the best term, as cognition gives immediately the anthropomorphic idea of human cognition. No, they mean by cognition the interaction of the amoeba, of the butterfly, of the dog with the environment, such that the living things chooses from the environment all what is necessary for its existence. So this little thing taken up by the amoeba or by the butterfly or by the man are part of life itself. And actually we can say with the word of the biologist that the organism creates from the environment its own world by this act of choosing, by this act of cognition. Now, create, it's a very strong word. But if you look at biology, you have many elements to say that this is indeed so. If you look at the spider, well, this environment, which is very specific, is created by it. But there is something more. He creates the environment, but the environment permits its being, permits the spider to be what it is, permits its life. So that there is this interesting dual relationship. This makes that, and that makes him. And this you find in biology over and over again. The termites make their own environment, but then the environment permits their existence. So it's for the ants and beavers, and but this is also for people. Mankind make their own hospital and churches and highways and hospitals, and all this environment permit the mankind its own existence. Again, this kind of double errors. Men make that, and that permits the being, the existence of man. So that we arrive at this point that there is a kind of co-emergence between the spider and the environment, between mankind and the environment. Co-emergence meaning that they fuse together to give a higher degree of higher level. And this emergence is due to the specific recognition. The amoeba recognize the sugar, and mankind recognize, whatever, oxygen, spaghetti, and the churches, and not other things. And so that we can now recognize, first of all, that this cognition is a continuum. So it starts, if you want, from the amoeba, and as you increase the complexity of the organism, so does the complexity of the sensorium, which interacts with the environment. At a certain point, you put little flagellum, or little fingers, or eyes, or then at a certain point you develop brain. But it's always the same mechanism of cognition, of intelligent, specific interaction with the environment. So then we have detected then three parts, and this trinity makes really what we call life at large. There is the biological structure. However, this has to interact with the environment to get food, energy, and to build a unity. As would not have sense to talk about mankind without the environment. And this interaction, this co-emergence, is made possible by this mechanism of recognition of what is good and what is not good. So that we have this kind of trinity to define life. And for mankind, then, this cognition may take the form of mind, perception and intelligence, perception and consciousness, and this brings to the notion of, which is very important today in cognitive science, of embodied mind, first proposed by Varela at the end of the 90s. Embodied mind meaning that it has no sense to talk about mind or consciousness as a kind of abstract term. This mind must always be participating into a physical structure, and vice versa. If you want to talk about life of human or any other living things, it has a sense only if this living has a kind of cognition, so interaction with the environment. Does not have to be a brain to have cognition. You don't need the brain, but you need the sensorium to perceive the environment. And your evolution has been such that the sensorium you have is specific for your species and specific for the particular environmental connection that you have. So that we are at the end of these stories, having and it's possible to summarize all what I've said in this kind of broad spectrum, where we started from the Big Bang, we formed the Earth on the crust, we had the formation of molecules also coming from the space, and due to a series of processes which we did not discuss here today, self-organization, emergence, we arrive at a more complex molecular structure, structure which were capable of making copies of themselves. From here, Darwinian evolution starts with the biodiversity, which includes human life. And human life is then characterized by brain, and with that certain degree of perception, intelligence, up to consciousness. And now this is the next step, as kind of byproducts of this consciousness, which is the capability of reflecting upon itself, himself, come out as self-generated values, things like moral laws, finality, meaning of life, and the notion of God. So that here in this picture, the notion of God and spirituality is seen as a kind of development of the immanence of the autopoietic machinery of producing complexity. So God seen as the product of the mind of mankind. I know that particularly when I used to give this lecture to younger people, there was some discomfort about God being the product and coming at the end. So then I learned to modify that a little bit, or to present an alternative in which, to make some of these younger people happy, I put God at the very beginning, as some people may like to think that God is the origin of the Big Bang and everything. And so if you want to put God here or God there, is in fact your choice, and you have all your life to decide upon the two possibilities. Thank you very much.